All right, let's just jump right into this explainer. Today, we're going on a microscopic journey to figure out what fundamentally makes something alive. I mean, we interact with the biological world literally every single day, right? But today we're going to peel back those macroscopic layers and look at the absolute foundation of existence itself.
Seriously, just look around at the living and non-living things in your environment. Have you ever actually stopped and wondered what exactly separates the animate from the inanimate? Like, what is it that a rock or a piece of plastic completely lacks that a plant, a bacterium, or, well, you possess? It's a deceptively simple question, sure, but the answer absolutely revolutionized our entire understanding of the natural world.
Which brings us to Section 1, the basic unit of life. And the answer to that profound mystery lies in a tiny, microscopic structure, the cell. Every single organism is made of them. Some just navigate the world totally on their own, while others, like us, are these vast, incredible collaborative networks of billions of cells. But the absolutely crucial point here is that the cell is the fundamental structural and functional unit of living organisms. Anything less than a complete cell, it simply cannot survive independently. By studying the chemical and physical processes inside this tiny unit, which is a super fascinating approach known as reductionist biology, we can actually start to understand massive physiological processes like digestion, memory, and even our own immune defense.
Moving right along to Section 2, the cell theory. To really grasp how this tiny building block creates life, we kind of have to look back at the historical journey of its discovery. And honestly, this wasn't an overnight revelation. It took decades of work. Back in 1838, a German botanist named Matthias Schleiden noticed all plants were made of cells. Literally a year later, zoologist Theodor Schwann spotted this really thin outer layer on animal cells, which is what we now know as the plasma membrane. Schwann boldly proposed that all plant and animal bodies are built from cells. But that begged the question, right? Where do they actually come from? Well, in 1855, Rudolf Virchow dropped a famous Latin phrase, omnis cellula e cellula, meaning new cells are exclusively born from pre-existing ones. That totally locked the cell theory into place. And our understanding has been evolving ever since, all the way up to Singer and Nicholson's famous fluid mosaic model in 1972.
So, let's get into Section 3, a microscopic overview.
Now, before we zoom all the way in on that cellular boundary, we really need to take a second to appreciate the sheer, astonishing scale of cells in nature. I mean, they drastically vary in size. On one end, you have mycoplasmas, the absolute smallest, measuring a mere 0.3 micrometers. Then, typical bacteria are slightly larger, maybe 3 or 5 micrometers. Scale up a bit more and you get human red blood cells at about 7 micrometers. But on the extreme other end of the spectrum, the largest isolated single cell in the entire world is the egg of an ostrich. Yeah, no joke, an ostrich egg is a single cell that you could, technically, scramble for breakfast.
But this immense diversity isn't just about size, it's also about shape. Cells are definitely not just uniform, perfect little spheres. They can be disc-like, columnar, thread-like, or just totally irregular. Why? Because a cell's form is deeply, deeply tied to its job. For instance, a long, branched nerve cell needs to transmit signals across vast distances in your body, whereas a biconcave red blood cell has to literally physically squeeze itself through your tiny capillaries. Form always follows function.
That brings us to Section 4. The fork in the road, prokaryotes versus eukaryotes. If we take a step back and divide the entire biological world into two distinct camps based on their architecture, we hit a massive fork in the road. Over on one side, we have prokaryotes. Think bacteria and blue-green algae. These guys are smaller, rugged, they multiply incredibly rapidly, and they completely lack a defined nucleus. Their genetic material is essentially just naked DNA floating around in the cytoplasm. Then, on the other side, we have eukaryotes. That's us, plants, animals, fungi. These cells are highly organized with extensive internal compartmentalization, a well-protected nucleus, and these specialized membrane-bound organelles.
Let's zoom in a bit closer on the prokaryote first. You can think of a prokaryote kind of like a highly efficient single-room workshop. It doesn't have internal walls, but it is heavily protected from the outside world by a tight, three-layered armor known as the cell envelope. The outermost layer is the glycocalyx, which can either be a loose slime layer or a really tough capsule. Right under that is the cell wall, providing strong structural support so the bacterium doesn't literally burst. And finally, on the inside, there's the selectively permeable plasma membrane. Now, even though these are three distinct layers, they work together perfectly as a single, highly effective protective unit.
Moving to Section 5, eukaryotic cells.
So if a prokaryote is that rugged one-room workshop, the eukaryotic cell is more like a bustling, heavily compartmentalized city. But interestingly enough, even within this eukaryotic city, there are different factions. Plant cells, for example, build rigid exterior walls, they utilize solar-capturing plastids, and they contain this really large central vacuole. Animal cells, on the other hand, completely ditch those rigid walls and plastids, but they bring their own unique structures to the table, like centrioles, which are absolutely essential for cell division.
Inside this eukaryotic city, the compartmentalization is just incredible. You've got the nucleus acting as the main command center, the mitochondria generating the power, and the complex folded membranes of the endoplasmic reticulum handling manufacturing and shipping. And we can't forget the cytoplasm. It isn't just empty, dead space. It is the main fluid arena where the chemical reactions happen that actually keep the cell alive. It's where the magic happens, really.
All right, our final section. Section 6, the boundary of life, the fluid mosaic membrane.
For our final act today, we are going to zoom right to the very edge of these cells. This brings us to the famous fluid mosaic model. Now, before the electron microscope, we basically just had to guess at what this structure looked like. But today, we know this boundary isn't some static, solid brick wall. No way! It is a dynamic, moving sea of lipids arranged in a bilayer. The lipid tails are hydrophobic, meaning they are literally water-fearing, so they point inward, safely tucked away from the watery environments on both the inside and outside of the cell. And floating around within this quasi-fluid sea is a constantly shifting mosaic of proteins.
Now, the exact chemical makeup of this fluid border actually varies depending on the cell type. But to put some numbers to it, if we break down the membrane of a human red blood cell, the biochemical data shows a super fascinating split. It's about 52% protein, 40% lipids, and 8% carbohydrates. That highly specific, balanced ratio is exactly what gives your red blood cells the flexibility they need to travel through your veins, like right at this very second.
All of this fluid structure builds a really vital, selectively permeable border checkpoint for the cell. It's all about transport. It allows certain molecules to just drift across without expending any energy whatsoever, which we call passive transport. Neutral solutes simply diffuse from an area of higher concentration down to a lower one. And when water does this, we call it osmosis. But because the inside of that lipid bilayer fears water, polar molecules can't just slip on through unnoticed. They actually need specialized carrier proteins, acting like molecular bouncers, to facilitate their journey across the border.
By looking this closely at the cell, we uncover the fundamental mechanical and chemical rules that govern all biology. But honestly, it leaves us with a truly provocative thought to end on. How exactly do these tiny, passive lipid bilayers, these microscopic transport mechanisms, and these floating proteins all aggregate together to create the endlessly complex behaviors of the macroscopic life we experience every single day? Like, how do these exact chemical reactions build memory, immune defense, and even human consciousness? That is the continuing, grand mystery of biology, and for sure, the true power of understanding the cell.